You'll be removed and you can rejoin. So not everybody, especially the student that just read the name. Okay. So, chalo. So now, yeah, where was I? I was talking about the breathing process. So why do we need? Why do we breathe? We breathe for a particular process where, while breathing, we take the atmospheric air, and this air is where our red blood cells they fix this oxygen or pick up this oxygen, go to the tissue, deliver to the tissues, and tissues somehow use this oxygen. Okay, for the process of respiration. So, in a very generic sense, what is respiration? Respiration is a process of production of energy, as simple as that. Now, if you see the significance of a particular thing, for example, we just told oxygen is very important for process. Oh, 1 second. Oxygen is very important for process of respiration. I just mentioned. Now, if you see where the oxygen comes in place with the respiration, you'll be very, very surprised. Okay, we'll talk about very interesting things. So, in higher organisms like animals, if you see, we have a very complex. Okay, if you see in vertebrates, especially in mammals, we have a very complex physiology to support this process. Energy is a very, very important criteria for all the biochemical cellular functions that happens in our body. So, if you see what kind of activities do take place, look at the amount of biomolecules your cells will synthesize. Look at the amount of growth and repair and reproduction your cells will undergo. Look at the amount of energy that is being spent to keep your body warm or hot or cold or sweat, whatever it is. Okay. So, for all this, the physiology requires this energy. So, what have these organisms figured out? We don't have a battery or we don't have a recharging port. So, instead, what we do is we have understood the way of storing this energy in the form of chemical bonds. So, we know that the very basic energy molecule or the source of energy molecule is a carbohydrate. And in plants, it is stored as starch. And then again, the starch is converted into glucose. In animals, it is stored as glycogen. So again, it is converted into glucose. So glucose becomes a very basic substrate, I can tell. So glucose becomes a very basic substrate for the process of production of energy. Correct, right? So what happens to this? The process is very, very simple. We have a complex molecule of a glucose, and within this glucose molecule, we have chemical bonds. So now, if I make this glucose into simpler forms, like when I break this complex structure into simpler structure, when I convert this, what happens is energy is released. So, okay, which is catabolism. So if I catabolize, okay, if I catabolize glucose, what do I get? I get energy. Now this energy, I cannot waste it, but instead I can create this into currency like money. Okay. So how do I convert this energy? I just convert this ADP plus phosphate to make it into ATP. The basic currency of energy is ATP molecules. Now, why do we store it in the form of ATP molecules? When I create bonds, the energy is stored in the form of bonds. And for any physiological process, my body knows to break these bonds, utilize this energy, and perform the following activities. Correct, right? Now, if I take in comparison between a plant and an animal, one thing that is very, very similar is we both breathe, we both respire. Two separate process, but two interlinked process, I can tell. So breathing is nothing but it's a mechanical process where I just inhale and exhale. What happens in the plants? Do plants inhale and exhale? Definitely they do, but we call it as the process of gaseous exchange. So in plants, what happens is not all the parts of the plant are equally participating in the process of respiration. If you just take an idea or if you just look into a plant and just imagine what is the plant physiology, we know that we have all the green parts of the plants which participate in the process of photosynthesis, where it manufactures our glucose. Whereas if you look at the same parts of the leaves, you can see that the leaves are embedded with minute pores, which we call it as stomata, which helps in the gases exchange. So here the leaves are very, very, what do you tell? They are very efficient for this process of, producing the glucose as well as getting the gas they need, or the air, yeah, the gas they need for the following processes. It's not the same in every part of the plant. For example, take the non-photosynthetic parts of the plant, such as a stem, such as a root. Okay, so if you take stems and the root, they don't participate in photosynthesis. They require support. How do they require support? Now the food is prepared in the leaves. Now prepared food have to be translocated to the non-photosynthetic parts, which the phloem does this job very effectively. So the glucose that is produced is converted into sucrose in the plants, in the plant leaves itself. From the sucrose from the plant leaves are translocated to different parts of the plant, okay, such as roots and stem, which are non-photosynthetic. Now, once we have the food reserve, we know that the very important component that we require to convert this food into energy is oxygen. So that is where the breathing comes. So for the breathing process, especially in the plants, what happens is, if you see in one plane, you have leaves, and the leaves are having this proper, design where they have stomata and the gases exchange can be perfect. Whereas if you see the other regions of the plant, such as stem, such as roots, how do they breathe? Do they really breathe, or how the gases exchange happens? That's a question mark. Of course, they breathe, but they breathe through some of the openings that are present within this bark or woody regions, which we call it as lenticels. So the lenticels in the non-photosynthetic part, especially in the stem, they participate in the breathing process. Whereas in the photosynthetic part, such as leaves, the stomata helps in the gases exchange. One of the fascinating things that we can understand is the plants do not require as much as breathing that animals do. See, we have to continuously breathe, whereas the plants don't have that obligation for many reasons. See, if you observe the process of photosynthesis, you know that the byproduct of the photosynthesis was glucose, oxygen, correct, right? Glucose and oxygen was a very important one. This oxygen is very, very important for the process of respiration. Okay, and plants give out oxygen as a photosynthetic byproduct. So that means they don't have to completely depend upon the atmosphere for the oxygen concentration. They just have it within their cells, and they can redirect this oxygen for their process of respiration. That is the one major advantage. So if you look at the plants and if you compare it with animals, plants do not require as much amount of supply of oxygen as the animals. As the animals, they require very less amounts. Okay, did you understand this? So this breathing process actually happens at a very specific space in a very higher rate. So, for example, during the process of photosynthesis, okay. So during the process of photosynthesis, what happens is there is a very large or high rate of gas exchange that is happening. Okay. So at this point, the respiratory rate is also very, very high. During the non-photosynthetic process, usually the gases exchange is very, very less because the stomata gets closed and they have to depend upon a very limited amount of gases exchange that is happening, and the energy production also drops during this process. What are we trying to talk here? We are just trying to understand that plants are no different from animals. Like how we breathe, plants also breathe. But breathing in the plants happens through the spores that are present in the stomata, in the leaves in the form of stomata, and in the woody regions like roots and the stems in the form of lenticels. Okay. And there are other supports, such as you can see that there are the parenchymatous tissue are very loosely arranged in the plants. So the loose arrangement of parenchyma also allows some of the gases to be exchanged. Okay. So this gas exchange, the breathing is one process that we have to understand, and it's not so complex. Okay. So next, coming to what happens after we breathe, the whole process or the whole idea of respiration is we take a complex molecule such as a glucose, and then with the help of oxygen what we produce is the energy. Okay. So this energy, as I told you, when you break a complex molecule into simpler molecule, now the energy is released, and the release energy is stored in the form of chemical bonds. Usually the major or the very common form of a currency of energy is ATP molecules. And with the waste of respiration, you can tell the byproducts of the respiration would be carbon dioxide and water. So this is a very simplest way to put or to understand what is the whole gist of respiration. Correct, right? So now, when it comes to the photosynthesis and the link with the respiration, as I've told you before, the photosynthesis we produce glucose, and in the respiration we utilize glucose. Okay. So now, if I look at organisms, what do organisms do? See, in the last class I told you, while studying respiration, you can really understand how organisms have evolved during the course of time since the life has begun. If you look at the life on Earth when it started, or in the initial phase of when the life was like blooming on this planet, what was not there was oxygen. So there was no oxygen in the environment or atmosphere. I'll write it as oxygen was not at all there in the atmosphere. So organisms had to find out some way to break down this glucose. And they did it anaerobically. So what am I saying, kids? Nothing. All organisms had this glucose, okay? And they didn't have oxygen in the atmosphere. So they had to find out some way to break down this glucose. And they did break down this glucose anaerobically or without the help of oxygen. So whenever a process happens without the help of an oxygen, it is called as an anaerobic process or anaerobic biochemical reaction or non-oxygenic process. So for this process, we don't need oxygen. And this process happens in the cytoplasm. Okay. And this process happens in the cytoplasm of all organisms. For example, it happens in the prokaryotes, it happens in the eukaryotic unicellular organisms, or it happens in the eukaryotic multicellular organisms. In all of us, it happens. Okay, which process? The glycolysis process. Okay, what is a glycolysis process? Glyco means glucose, lysis means breakdown of glucose. So we'll talk about how the glucose is broken down, but initially, what we have to understand, glycolysis is a strictly anaerobic process. And where does this happen? It happens in the cytoplasm. So the explanation says that since in the beginning of the life there was no oxygen, and life adapted to break down this glucose without the help of an oxygen, and that was the origin of the glycolysis. Glycolysis is also called as an EMP pathway based on the three scientists that contributed in breaking down this cycle. Okay, so to understand the glucose catabolism, I can tell you why, because we are breaking down the complex into simpler forms. It's very easy to understand and it is very fluent to understand. Okay, so what happens in glycolysis is it's very simple. First, we have 6 carbon atoms in a glucose molecule, and then we break it down into 3 carbon atom containing compound called as pyruvate. Okay, glucose is broken down into pyruvate. So this process of breaking down excuse me, just give me a second. Sorry, people. Hold on, I'll just open another PPT so that it will be even better. One more. Hold on, people. So just for this slide, I need this slide. So sorry, where did it go? Yeah, take a look at this particular process. Okay, hold on. What's this? Yeah, yeah. Take a look at this particular process. Okay, I hope you can see the letters. If you cannot see the letters, please let me know. Okay, it's very important. Yeah. So is it visible? Are the letters visible, students? Now, little bit. All right. Okay, so if you look at a biochemical process like glycolysis, we can call glycolysis as a multi-step process. Uh-oh. Okay, it is called as a multi-step process. Okay, so for any process to be accomplished, what is required is energy. And our cells are very, very super smart, and especially organisms are very smart. And you can tell stingy, or you can tell that conjus in common language. Why? Because they don't want to spend more energy here. We want to conserve energy. We want to save as much as possible. And for that, they use or they take the help of complex varieties of enzymes that participate in a series of chemical reactions, thereby helping the glucose break down without spending much of an energy. Without spending much of an energy. So these enzymes are very, very important for us. Understood? So let us summarize the process of glycolysis stepwise. So if I look at the plants store their energy in the form of starch. Okay. What happened to this? Wait, students, I'll just have to annotate. All right. Hold on. Sorry. This thing's gone crazy. Where is it? Yeah, here it is. Nope. Wait, I will do this now. Yeah, we are it. Sorry. Yeah. So if we summarize glycolysis, or we have to understand glycolysis, as I told you, it's a multi-step process. That is the first thing, and it involves several enzymes that help in this accomplishment of process. And the reason is they want to spend less energy as possible. Okay. So let's see how it happens. In the plants, what happens is it starts from starch. Okay. Arey yaar. Okay. Is right. Yeah. Erase. Yeah. It starts from starch. Uh, my goodness, everything is stopped. Excuse me, students, just a minute. Okay. The PowerPoint closed. I don't know why. Okay, let me open. Sorry about this disturbance. Okay, usually it doesn't happen, but it's strange it's happening today. Come on. Okay, until it opens. Are you guys able to follow? Yes, no. Right. Okay, we have it. Let's get back. Okay. Chalo. So it starts from sucrose. Hope it writes. Come on. Yeah. It starts from sucrose. Okay. The sucrose is converted into glucose. All right. The stored food in the plants is in the form of sucrose, and the sucrose will be converted into glucose. Okay. Now, what happens is once we have glucose, you just have to understand that glucose is converted into glucose 6 phosphate. It's simple. In glucose, we have 6 carbon atoms, isn't it? 1, 2, 3, 4, 5, 6. You see, glucose is converted into glucose 6 phosphate. At the sixth carbon atom of the glucose, a phosphate molecules get added. Very simple. Okay. Now, this particular process, okay, hold on a second. I missed something. The sucrose is converted into glucose with the help of an enzyme called as hexokinase. Okay, just keep that way. So this hexokinase is very, very important. So hexokinase help in the conversion of glucose. That's done. Now, once we have a, yeah. Once we have the glucose, what happens for a glucose molecule at the sixth carbon atom, a phosphate molecule gets added. How do we get this phosphate molecule? For that, we have to invest. So if you can if you can see the whole picture, in the whole picture can be divided into two important phases. One is you invest, and the second thing is you get the returns. Investment is that the cell is spending some energy, okay, or some factors, so that is called as investment phase. Whereas in the second way, after the investment, what do we get? We get the returns of it. So we have the investment phase and then we have the return phase. Now we are looking at the investment phase. So in the beginning itself it happens. What happens here? We have a glucose molecule that contains 6 carbon atom, and in the sixth position a phosphate molecule gets added up. Okay, and this phosphate molecule gets added up with the help of an enzyme called as hexokinase itself. Okay, hexokinase. Is it clear? So that is the first thing. Now this glucose 6 phosphate, okay, so I'll write it here, glucose 6 phosphate in the presence of, see, this glucose 6 phosphate in the presence of another enzyme called as glucose isomerase, you can also call it as, or you can call it as phosphoglucose isomerase. What it does is, now we have glucose. Glucose was converted into glucose 6 phosphate by addition of a phosphate molecule to the chain by investing an ATP molecule. So what did we do? We invested in ATP molecule. Why did we invest in ATP molecule? ATP that was invested, what this hexokinase does is it will break this down into ADP plus phosphate, ADP plus phosphate, and that is how the glucose gets the phosphate. You understand my point? So glucose got the phosphate by the investment of one ATP molecule in the presence of hexokinase, and then this phosphate molecules comes and attached to the sixth carbon atom. Thereby, glucose becomes glucose 6 phosphate. Done. So the next step. What is the next step? In the next step, we have glucose 6 phosphate, and the glucose 6 phosphate is isomerized. So what is isomerization? When glucose 6 phosphate is converted into its geometrical isomerism, that is fructose. Correct, right? We can see that glucose and the fructose are the same formula, but their structure is different, which is called geometrical isomerism. So for that, an isomerase enzyme will come. The name of the enzyme is very relevant to its function. So what happens here? there is something called as phosphoglucose isomerase. Okay, phosphoglucoisomerase. What it does is it converts glucose 6 phosphate into fructose 6 phosphate. Easy. First glucose, then we have glucose 6 phosphate, then we have fructose 6 phosphate. We got the fructose 6 phosphate. Again, we have to go for investment. What is happening here? Now we know that the fructose 6 phosphate is also like glucose, which was having 6 carbon atoms as usual. 1, 2, 3, 4, 5, 6. Correct, right? Now we know that in the beginning itself, we added one phosphate molecule at the 6th position. That is why it got the name glucose 6 phosphate. Then it was isomerized to fructose 6 phosphate. Now assume that you are looking at the structure of fructose 6 phosphate. Now what I'll do is I will invest one more ATP, not I, the cells. They'll invest one more ATP, where ATP will be converted into ADP plus phosphate. And this, whatever the phosphate they get, they will try to attach it to the first carbon atom. Okay, now you see the first carbon atom also have phosphate, the 6th carbon atom also have phosphate. So for this process, another enzyme, so this enzyme is called as fructokinase 1. Okay, it's okay. Fructo, hold on, fructokinase is the enzyme. What does this fructokinase will do? Fructokinase uses the ATP. It breaks the ATP into ADP plus phosphate, thereby converting fructose 6 phosphate into fructose 1 6 bisphosphate. Two phosphates are there, no? That's why it becomes fructose 1 6 bisphosphate. I'll write it as FRU, fructose 1 6 bisphosphate. Okay, you have to remember in the very first 4 steps itself, we have we have invested 2 ATP molecules already. Now we have fructose 1 6 bisphosphate. The fructose 1 6 bisphosphate will be broken down. Okay, how it will be broken down? I will show you now. Okay, very simple. I hope you're understanding. If you're not understanding, you're very free to put it in a chat box and we can find ways to understand. Okay, no problem. Just give me a second. All right, students. So where are we now? We are here. We have carbon, 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 carbon. So 6. In the first position, we have phosphate. In the 6th position, we have phosphate. And we call it as fructose 1 6 bisphosphate. To this fructose 1 6 bisphosphate, an enzyme called as aldolase comes. Okay, an enzyme called as aldolase. What does this aldolase do? Aldolase will break down this glucose molecule, that is the fructose 1 6 bisphosphate, into 3 carbon containing compounds. So what are these 3 carbon containing compounds? One thing, it is called as dihydroxy, okay, acetone phosphate, dihydroxy acetone phosphate. So see, this dihydroxy acetone phosphate is interconvertible. You understand this point? See, I will put 2 compounds. One is dihydroxy acetone phosphate. Other one is glyceraldehyde 3 phosphate. Okay. Let's assume that I have one molecule of fructose 1 6 bisphosphate that is having 6 carbon atoms. If I break a 6 carbon atom containing molecule into half, what do I get? I get 2 chain, right? One 3 carbon containing molecule, other one 3 carbon containing molecule. So what happens is, let's say that I get dihydroxy acetone phosphate. This dihydroxy acetone phosphate can be converted into glycerol 3 phosphate. And this conversion happens through another isomerase. This is isomerization reaction. It's the same formula but different structure, as simple as that. So what happens? You have triose phosphate isomerase. Okay. Now what it does is it converts this dihydroxy acetone phosphate into glyceraldehyde 3 phosphate. That means at the 3rd position you have a phosphate. And how many molecules do we get? Let's say that I have fructose 1,6 bisphosphate. I break it down. I get 3, 3. Correct, right? 6 divided by 2 is 3. So I get 3 and 3. So let's say that we have two molecules of dihydroxyacetone phosphate. Now what happens is by isomerization reaction, I will be ending up with two molecules of glyceroldehyde 3 phosphate. Understood? Once I get glyceroldehyde 3 phosphate, okay, now what happens is you have the reduction process. Hold on, very important step where the NADP plus comes into the play. Okay, so here NADP plus is, okay, hold on. NADP plus is oxidized to, is reduced to NADPH. So now what happens? I have glyceroldehyde 3 phosphate. Glyceroldehyde phosphate 3 phosphate is converted into 1,3 biphospho or 1,3 bisphosphoglycerate. See, when I say 1,3 bisphosphoglycerate, you already have an idea. Before, what happened? I have 3 carbon atom, glyceroldehyde 3 phosphate. At the third position, there was a phosphate molecule. Correct? Now, what is happening? There is one more thing that is coming, that is called as NAD, NAD plus, and NAD plus plus another phosphate. Two phosphates are coming together to reduce this NADP into NADPH plus H plus. So when this is happening, what does this lead to? This leads to addition of this phosphate molecule into the first position. Correct, right? Now we have 1, 3. So that will give 1, 3. How many are there? 2. That is why I call it as bis phospho phosphoglycerate. Okay, 1,3 bis phosphoglycerate. And again, it is an enzymatic process. Conversion of glyceroldehyde 3 phosphate into 1,3 biphosphoglycerate happens through an enzymatic process. And which is our enzyme? It is called as glyceroldehyde 3 phosphate dehydrogenase. Okay, reduction process happens. That's why we have dehydrogenase. Clear? So where are we? What are we happening? There is a question. Where is NAD plus invested? NAD plus is invested to convert glyceroldehyde 3 phosphate into 1,3 bisphosphoglycerate. Done, people? So once we have 1,3 bisphosphoglycerate, now what happens? Now we get the return phase. Now, how do we get the return phase? It is so logical to understand this. Okay, very simple. See, people, I have two phosphate molecules. Okay, I will knock out one phosphate molecule. Now, what does this lead to? My compound here was 1,3 biphosphoglycerate. Now, if I take out one phosphate, what it will become? 3 phosphoglycerate. At the third position, one phosphate is there, right? It will become what? 3 phosphoglycerate. Now, what happened? One phosphate is gone. What do I do with this one phosphate? I will add it to the ADP. ADP plus phosphate will give what? ATP. Is my ATP an energy molecule? Yes. So where do I get this investment? Here. Okay. Here I get the returns. One molecule of ATP I get. All right. So next, what happens? 3 phosphoglycerate is reduced to 2 phosphoglycerate. That means one more carbon atom gets removed, and 2 phosphoglycerate will be converted into phosphoenol pyruvate, and eventually phosphoenol pyruvate will be converted into pyruvate. Now remember, when we broke down our 1,6 biphospho, wait, hold on, let me make it clear for you. We have two phases here. One is the investment phase, other one is the return phase. What are we doing here? We had glucose. Glucose was converted into glucose 6 phosphate by addition of one phosphate. To add one phosphate, we need one ATP molecule. Simple. Again, glucose 6 phosphate is converted into fructose 6 phosphate by isomerization. No energy is required. Isomerase will come and help here. Now, fructose 6 phosphate is converted into fructose 1,6 bisphosphate. For that, we need one more phosphate. That's why we add one more ATP here. So, two ATP molecules gone. Now, what happens? Each 1,6, fructose 1,6 phosphate, what it can do is it can be broken down into 3 carbon containing molecule. So what do we get when we break down fructose 1,6 bisphosphate? We get glyceraldehyde 3 phosphate. So if I break one molecule of fructose 1,6 bisphosphate, How many molecules of glyceraldehyde 3-phosphate I get? Two molecules of glyceraldehyde 3-phosphate I get. Correct, right? So this glyceraldehyde 3-phosphate is again added one more phosphate molecule. This time not by ATP, but by the reduction process of NAD plus. I take NAD plus with the help of two phosphate molecules. What happens here? I get NADH plus 2H plus. What happened to this phosphate? The phosphate got added to our glyceraldehyde. Now, what do we get once we add the glyceraldehyde? One more phosphate. Here it was only one phosphate, that is glyceraldehyde 3-phosphate. Now I get glyceraldehyde, that is 1,3 biphosphoglycerate. Okay, or glyceraldehyde 1,3 biphosphate is also correct. Okay, so 1,3 biphosphoglycerate I get. Now again I start removing this phosphate. 1,3 biphosphoglycerate, I take one phosphate molecule out, I get 3-phosphoglycerate. 3-phosphoglycerate is converted to phosphoglycerate. 2-phosphoglycerate is converted into phosphoenol pyruvate. Phosphoenol pyruvate, okay. See, when I have 2-phosphoglycerate, a water molecule is removed. Once it is dehydrated, phosphoglycerate is converted into phosphoenol pyruvate. Eventually, it gives me pyruvate. So, if you have to understand it, it's very simple. Here the equation changes. Where here it was only one molecule of fructose 1,6 phosphate, bisphosphate. With that, I will get two molecules of glyceraldehyde 3-phosphate. With that, same thing, two molecules of 1,3 bisphosphoglycerate, two molecules of phosphoglycerate, 2,3-phosphoglycerate, two molecules of phosphoenol pyruvate, and yeah, same thing, and two molecules of pyruvate. I think it's very easy to understand how we got this two from one, because it is 6 carbon atom molecule. Here it is 3 carbon atom molecule. Clear? Yes. So now I would make it simpler for you. All these enzymes you need not remember. What you have to remember is hexokinase. Very important. What do hexokinase do? It helps in converting glucose into glucose 6 phosphate. That's it. Okay. Rest of the things is how many ATP, how many ATP is invested, how many ATP we get returned. Let us watch. Okay. In the first stage, in the investment phase, what are we doing? In the first stage, we put one ATP molecule here, and again we add another ATP molecule, correct? And then what do? We invest two molecules of NAD plus two molecules of phosphate. That is not okay. That's okay. But you have to remember this, two molecules of NAD plus. Now what do we get? We get in the first phase here, we get two molecules of ATP. Now don't ask me how. You know that there are two 1,3 biphosphoglycerate. For every 1,3 biphosphoglycerate that is converted into 3-phosphoglycerate, we get one ATP. So since there are two, we get two ATP. Again, we get two molecules of ATP here. Correct, no people? So totally how much? We get 4 ATP. How much have we invested? 2 ATP. ATP, 1 ATP, 2 ATP. So 4 minus 2 would be 2 ATP. So this partial breakdown of glucose through glycolysis process, eventually glucose is converted into pyruvate, will result in the formation of two ATP molecules plus two molecules of pyruvate. As simple as that. So this process is called as glycolysis, and it happens in the cytoplasm in all organisms. Did you guys understand this process, or you want me to repeat any part of it? Yes, no? Students, are you there? We almost spent an hour talking about glycolysis. Okay, so it becomes easy okay once we move on. Is it understood? Just give me an assurance that I can move further, or should I repeat? All right. So nothing. You did not get it. Should I repeat? Audio is not clear. Please increase the audio. I'm sorry, I didn't see your message, but I hope it's okay. All right, students. So I will move further until okay. Kundalal Singh. I hope I'm pronouncing it right. And then we have Ebin. You guys said that audio was not clear. I hope it is clear now for you. Yeah. All right. So students, please learn to interact. Okay. So anyways, you'll get there as we move further. No doubts. So once we have glycolysis and then we have the byproduct of the glycolysis is two molecules of pyruvate and two ATP is a very less amount of energy yield. When you compare to other process, glycolysis is very thoda energy we will get. So but it's not the end of the process. It's just we got it started. Okay. So now what happens to this pyruvate? The fate of the pyruvate is determined on the type of cell we are talking about. For example, it takes three parts basically. One, in anaerobic organisms. For example, you take yeast or you take any more unicellular organisms that are anaerobic or facultative anaerobes or obligate anaerobes. Facultative means sometimes they require oxygen, but they are anaerobic, and obligate are the ones who are compulsorily anaerobic in nature. So these are one thing, that is anaerobic part. Second part is in the aerobic organisms, there might be an anaerobic reaction. For example, I start working out, and then as I keep on working out, now my muscles get fatigued and tired, and I'm not able to supply sufficient amount of oxygen to my tissues. Now, my muscles go into an anaerobic state. That is the second thing. And the third one is the regular aerobic respiration. So first, let's talk about the two conditions. What is the two conditions? The pyruvic acid or the pyruvate that is released in our tissues or in our cells or in the bacterial cells, what happens to it? So first we'll talk about lactic acid fermentation, and then we'll talk about alcohol fermentation, and finally aerobic respiration. We have very detailed things to talk about. We'll talk about it. So in anaerobic reaction, especially, as I told you, if you take any fungi or a bacteria, this thing is possible. That is, alcohol fermentation is possible. In tissues where there is less oxygen for during the exercise and leads to lactic acid fermentation. How does this happen? Look here. We have glucose, right? Glucose was converted eventually into pyruvic acid by the following steps. Once we have pyruvic acid, now we have very important enzymes like alcohol dehydrogenase. Okay, is one of the enzymes that is used. So this pyruvic acid, in the presence of NADH plus H plus, what happens is NADH plus H plus is reduced. When this is reduced, the pyruvic acid is converted into ethanol plus carbon dioxide. Okay. Next one. In the same case, in our tissues, what happens? Again, the pyruvic acid, in the presence of NADH plus H plus, is reduced to NAD plus. So that will lead to the formation of lactic acid. So we have alcohol dehydrogenase, and then we have lactic acid dehydrogenase, which are the enzymes that help in this process. Okay. But here what happens is the conversion of pyruvate into lactic acid or ethanol is a very low energy process. So only 7% of the energy yield will get by breaking down of glucose and converting into alcohol or lactic acid. Understood? And basically, even the cells, for example, yeast, which produces the alcohol, it cannot accept unlimited amount of alcohol in its environment. It can only tolerate 13% of alcohol. See, as we metabolize during this metabolic reactions, byproducts are produced. For example, we breathe out and the carbon dioxide is coming, right? Similarly, when the pyruvate is is interconverted into lactic acid, or it is converted into alcohol, especially in some of the organisms like yeast, which produce alcohol in wines. So it cannot tolerate more than 13%. It will get alcohol poisoning and eventually cells will die. That is first thing. Second thing is we have very less energy production with respect to fermentation process, okay, which is only 7% of the total yield. Clear? So we have two important process. One is we have lactic acid fermentation and we have ethanol fermentation, which are the two important process that happens during anaerobic reactions. Okay, just give me a second. I have one more thing to discuss. Yeah, hello. Anyway, we are out of time. We'll take up in the next class. Okay. Do you guys understand? Yeah. Is it clear, guys? So let's pause it here and take the next topic. Usually aerobic respirations is a little bit complex. Okay. Hold on. Yeah. Aerobic respiration